Thanks, everyone. I have about 5 minutes of comments planned, meant to spur on more discussion, hopefully. So I want to talk a little bit about DNA sequencing and the opportunities associated with that. So this is the big hullabaloo. That's why we're here. That's what we care about. This is what's changed. Many microbiologists and biologists have sometimes divided biology to say 2005 and post-2005. What happened in that year? DNA sequencing became very inexpensive. If we were back in the post-2005 and we were making clone libraries, we would not be able to make as good of associations with epidemiology studies. We wouldn't be able to make analyze 12,000 samples in a hospital. We wouldn't be able to analyze 1,200 samples in a hospital, or if we did, it would be very difficult. This has opened everything up. And you can see an order of magnitude shift in the cost of DNA sequencing that came along with sequencing by synthesis type technologies, right? The advantage is that it's rapid, the sequences are smaller, but we're developing better ways to deal with that. Now, the problem that initially came with this was associated with Moore's law. We didn't really have the computational power, or we didn't have the computational tools to handle all these sequences. That's still a problem, but that's getting better and better and better. All right. How do we go down? Okay. So I've listed 5 or 6 things that we can do, some of us are doing, and things that I think we should be doing with all this DNA sequencing capabilities with reference to the indoor microbiome. So for bacterial libraries, to understand the diversity of bacteria, we can use PCR primers. We can target 16S RNA encoding genes for taxa or phylospecific communities or very broad communities. We can use ITS primers for fungi to look at those communities. There are no such ubiquitous genetic elements for viruses, but that's okay because we can just sequence a lot. And if you sequence a lot and you can assemble these things back, we can use a process called metagenomics, which is just pretty much sequencing everything and sorting it back out later. And we can identify viruses as well, right? qPCR gives us a quantitative sort of foothold in this. And then there are many, many other techniques that we could be using that really aren't being used in this community right now, that are associated with activity, that are associated with gene transcription, that are associated with looking at metabolic models of microorganisms. And I'll give you just one example. This is with viruses. This is something that I hope that we can do much more of. You know, we focus a lot on bacteria. We're focusing more on fungi. We don't focus on pollen at all, which I think is to our disadvantage. I think we should also be focusing more on viruses. This is an example. Unfortunately, this is in wastewater. We don't have such an example of this in the built environment. They're growing. But this is virus diversity. And I'll show you on the left. If you look at what you get when you sequence a bunch of viruses or virus-sized particles, you get a lot of stuff that's protein, but protein that hasn't been described yet, right? That codes for protein, that's unknown things. The other bulk amount you get are probably protein that can be or sequences that can be annotated. These are mostly bacteriophages, and then a small section of them can be human viruses. But if you sequence enough, a small section can be something significant. This was done in wastewater, and even in wastewater, with all the tradition and all the experience in wastewater, new insights were developed by using methods like this. We found things like papillomavirus. We found adenovirus very commonly. New viruses like classivirus. Coronavirus was much more common in human populations than we thought before. And things like Coxsackie virus and rhinovirus were much less common in populations. Imagine if we could take these types of techniques and apply them to the indoor microbiome. What would we learn about infectious disease? And I think that this is an approach, as well as the great approaches used by qPCR and computational modeling that are going on right now for infectious disease transmission, especially viruses in the indoor environment. I think this could be a very helpful tool, and I hope we can move much more into this area. Oh, okay. One last comment, and it's about applications. Because the audience here is a lot of engineers in public health, really the applications we think about are asthma, allergies, infectious disease, and a healthy microbiome that should have a Y there, right? The idea that our microbiome is also responsible for our good health as well as our bad health. We know much less about how the microbiome modulates our healthiness, but we know that there's something there, and that's something we should probably be thinking about. And then the design and operation of buildings to promote that healthy microbiome. Great.
Okay, thanks, Jordan. And then, did you have more there?
No, I don't know where that came from. This one was supposed to be my last slide.
Okay.